Hello, I'm Dr. Jimmy Bagley, and I'm a professor of kinesiology at San Francisco State University. I'm going to talk to you a little bit about our lab's research on skeletal muscle physiology, plasticity, and performance today. Like I mentioned, I'm at San Francisco State University. We've got a beautiful campus out here in the west side of the city. It doesn't always look like this. So if you've been to San Francisco, it might be foggy sometimes, but sometimes we do get this great view and you can even see the Golden Gate Bridge out there. If you're interested in joining us, we do have a really awesome group of researchers and professors that work in our Department of Kinesiology. We have a grad and undergrad program, and here's a link right there if you're interested in this stuff. So I like to start my talks with acknowledgments because I know a lot of this stuff usually gets caught till the end. So I just want to make sure to acknowledge our students for sure and colleagues. A lot of the students collected the data I'm going to share today. So I get to be the one to talk to you all about this cool stuff. And, they're the ones down in the trenches that are doing the hard imaging and exercise physiology work I'll get to talk to you about today. And then also our funding and support. Of course, we couldn't do this work without funding. So a lot of this has come from things like crowdfunding that we've done with our colleagues, as well as government, and then private foundations as well. And then key collaborators. There's a lot more folks than here, but some of the main universities that we've worked with on this stuff is Cal Poly Pomona, Cal State Fullerton, University of Arkansas, University of Kentucky, and the University of Southern Georgia. And then another big deal, it's kind of wild, time flies, but we just celebrated our lab's 10-year anniversary, the muscle physiology lab that I started at San Francisco State back in 2015. And so, again, a lot of this research has come from the years past, and just want to acknowledge those students before we get going. So, to talk to you a little bit about the research in the muscle physiology lab, is we look at the body kind of from the whole body all the way down to the molecular level and everything in between. So, basically, we can think of whole body physiology measurements as doing performance tests like VO2 max tests, like you can see here on the right, some of our students doing that, or even just lifting performance tests for strength or power or speed or go on the track, stuff like that. And we can break that down even further from the whole body and look at the body in the form of organ systems. And that way, look at the cardiovascular system, look at the neuromuscular system, musculoskeletal system, even the GI, all this stuff works together during exercise. And then we've got the cellular level. So we can get down, every system's got its certain cell types. So this is an image of skeletal muscle fibers, zoomed in about 20 times what your eyes can see. So you can see here, these things might look kind of strange. In this case, they're obviously outside of the body. They're actually clear and see-through. So it might not be like your textbooks where it's got these nice arrows pointing to the Z disc and all that. In real life, we can see some of those things, but we might have to label them and stuff. So this is an example of what muscle cells look like when you isolate them under a microscope. And then from there, if we want to get down even further, we can, like I mentioned, we can stain or label different parts of the muscle fiber. So this image here is red. The red part is actin. So if you look really closely, you can actually even see the lines in there. So you can see the actin filaments, and then blue is DAPI, and that labels all the DNA. And most of the DNA in there is inside the myonuclei. So that's going to label muscle nuclei. So that's the cellular level. But what if we want to get down even deeper, right? Down to things like the molecular level. We can look at protein content, using blots and stuff, gene expression, and then even things like the microbiome, and look at epigenetics. And the rabbit hole keeps going down and down and down, which is really cool. If you're interested in this stuff, there's never going to be something not to study. I don't know if I said that right, but you're always going to have something to study because there's so many things and it goes deep. So our lab again looks at the whole body all the way down to that molecular level, and then our main goal is to find out how to improve human health and performance. So very big goal, right? And we're just going to be a small piece of that big puzzle. Also, since probably several years ago, back in 2018, we started even working with virtual reality exercise gaming, and so we even incorporate things, technology, wearables, and stuff like that into our research as well. So the discussion plan for the next few minutes here is I want to talk about muscle cell plasticity with exercise. So plasticity, if you think about plastic, it's pliable, it's adaptable. We'll talk about that. We'll talk about hypertrophy, what some of our research and how muscle fibers and muscle cells grow. Atrophy is the opposite, how they might shrink or get smaller. And then metabolism and fiber type. So those kind of go hand in hand. Fiber type um, is related to metabolism, but also function. So that's what's kind of cool about it. And then I'll get into some of our future research direction stuff that's not even directly related to skeletal muscle, but everything kind of is. So I'll talk about that. And then I have a personal science story at the end talking about how a research idea can become a reality. So if right now you're watching this and you're, in high school or college, community college, undergrad, uh, grad school, PhD, and you don't know what's going to happen with research or it seems kind of intimidating, I'll have a little story about how I got into the research field here in a second. So this is a picture of a cross section of skeletal muscle. So if you look here, I mean, I think it looks really cool. Everything in the body looks so cool, right? Because it's got a pattern here, but everything's a little bit different. So each one of these is encircled by the basal lamina and parts of the membranes that go around the outside of the muscle fiber. The muscle fiber itself is dark because it's not labeled for anything. So those are those dark parts. And then the nuclei are there all around the muscle fibers. So that's what it looks like if we get a cross section. Um, if we think about muscle plasticity, we can think about things growing bigger or smaller like that, but we can also take a muscle fiber. And if you look here on the left side of your screen, this is a muscle fiber. Instead of looking head on, kind of flipped it and we isolated one fiber. And now we're looking at it what we call longitudinally. So you can see the muscle fiber, like I mentioned, it's labeled actin in red in this one, and then the myonuclei are there in blue. And this is a human muscle fiber. See, there's lots of myonuclei, which we'll talk about here in a second. So some of the main parts of plasticity that you can study are hypertrophy. So that would be muscle growth, like an atrophy, which is muscle loss. So these are kind of opposite pathways that work in opposition of each other. And then metabolism, I kind of consider the foundation of all of this. So the whole goal of metabolism is to break down what you eat, make ATP. ATP is the currency. Remember, the energy currency of the cell. You might hear about that in even high school biology. But our goal is to make ATP, and muscles use a lot of ATP, not just for movement, but also just to keep everything happy. So just even staying in homeostasis, like you say, everything just chill, right? You're still using a lot of energy just maintaining muscle mass. So metabolism is really important. This is a picture of a nuclei. I even zoomed in farther so you can see that they're kind of like, oblong or football shaped almost, but skeletal muscle nuclei are again everywhere in the muscle fiber itself, and they have a lot to do with hypertrophy and atrophy and growth. Let's start with talking a little bit about hypertrophy. The myonuclear domain is one thing that's been studied for the last couple decades, and people have been looking at it and how that relates to hypertrophy. And what that means is that you have all these nuclei around your muscles, right? So in most cells, think about how many nuclei you have. So most cells, you only have usually one nucleus. So you only need one set of all the DNA so you can, make mRNAs and those can go out and make proteins. But in skeletal muscle cells are huge. They're long. They could be, centimeters long, which is pretty big. And they need a lot of protein synthesis going on. They need to make a lot of protein. So it's more helpful to have more DNA in more places so that you can make copies or you can, make mRNAs and then those can go out and make proteins. So the idea here is if you have more nuclei, you could have more potentially protein synthesis down the road and more growth. So what happens is what people think and people have shown in research is that let's say you got four myonuclei around this muscle fiber. Instead of longitudinally, we flip it sideways. Now we're looking head on, kind of like pizza slices. Each one has its own myonuclear domain. So it's a theoretical area of the cytoplasm that, or sarcoplasm, same thing, that the muscle that each nuclei controls. We'll say that's what people think. Well, as the muscle fiber gets bigger, let's say the myonuclei have more space they've got to control or they've got to take care of, and it becomes inefficient enough to where satellite cells are brought in to make more nuclei. So you can actually make more myonuclei, add those, myonuclear addition, and then as the muscle fiber gets bigger and bigger, each area of the myonuclei that it can control is a little smaller. So that's the idea that at some point you're going to need to add more myonuclei to grow. Well, kind of like shifting gears, but staying on that topic with myonuclei, if we look at fiber types. So if you're unfamiliar, do a search, some of the research our group's done, or you can look at my colleague, Dr. Andy Galpin's got some great stuff out there on YouTube and other places, but talking about myosin heavy chain, that's a way that we can measure muscle fiber type. Usually people think of slow twitch, fast twitch. There's also multiple hybrid types in between and multiple types of fast twitch. So if we think about in humans, for example, we have a type 1, which is a slow twitch fiber, type 2A, which is a fast twitch, and a type 2X, which is even faster. Sometimes people call it super fast, or it's even faster than 2A. And then we've got these intermediate types that have multiple isoforms. Slow to fast is also oxidative to glycolytic. So we've got ones that are more suited to go longer for things like just postural support. So if you're sitting or standing right now, you're probably using part of your back muscles like the rector spinae. Those are mostly going to be these slow oxidative fibers. You should be able to stand for a long time. Might not be comfortable, but you can. But what about if you want to be jumping up and down and sprinting? You're going to be using more fast twitch fiber types, 2As and 2Xs, because those are going to fatigue faster, but you're going to be able to put out, more power and speed. And on that note, remember I was talking about the myonuclear domain size. Well, what we found, and what other groups have found as well, is that the smaller or the slow twitch fibers typically, in humans and in animals have a smaller myonuclear domain, meaning that they have more myonuclei per area of volume. And then it goes kind of in a stepwise fashion down to faster twitch fibers that have more space in between the nuclei, so they have less density of nuclei. We found about 20%. Other people have found similar, a little more, a little less, depending on the muscle and everything. But you can usually assume that, what's the purpose of that? Well, what people think is it might be that rate of protein synthesis needs to be higher in these slow fibers that are being used all the time and turned over all the time. And so they might have more nuclei in those areas to keep that up. So, that kind of makes sense. And again, that's a hypothesis for a lot of studies. Whereas fast twitch fibers, you're not going to use those like every minute of every day, so they might not be getting broken down as much or being used as much or growing as fast. So the rate of protein synthesis might need to be slower. It's probably more complicated than all this, right? But this is kind of the idea, and it kind of, again, does make sense. So this is the active area of research. Some of the cool stuff is that we're finding out is because the technology is getting so much better. So this is even like, I want to say, oh, this is an old microscope. No, we have new ones now, but this is still a pretty good confocal microscope down in our lab. This is a Zeiss. We have Leica. There's some really cool new super resolution confocal microscopes. And what is a confocal microscope? I'll give you just a quick rundown of what it is. It's basically I'm going to say a normal fluorescence microscope that's got some fancy gadgets on it. So most microscopes have a detector or a camera, what you can think of. Most have a light source like a laser. They got an objective lens. They've got a focal plane, things like that. We put a muscle fiber here. The difference between most microscopes and this confocal is that this has a confocal pinhole, or a pinhole that's small that you can change size. You can move that past the detector, and then it's got this mirror that can bounce certain wavelengths off and put certain wavelengths into the sample, and certain wavelengths don't hit it. And I'll show you what that means right now. So let's say you hit it with one wavelength, goes through, bounces back, goes into the detector. The reason why it's nice to have that pinhole is because any other wavelengths of light are going to get dispersed and not go into that. A normal one, you're going to get just light coming in all the time because you don't have a confocal pinhole. Well, then what this can do is move really fast across a sample and then inside a sample too, and get a 3D image of what it looks like. Really small, down to like the micron level. So pretty small, especially for muscle fibers. You can get really great images of that. Um, this is some research from one of our previous students, our alumni, Stephen Maychek, who got his degree here, I don't know, a few years ago, not that long ago, but then went on to get a PhD and now is down at CSU Monterey Bay doing research as well. But his master's thesis looked at muscle fiber type and morphology in some middle-aged elite powerlifter using anabolic steroids. And what we found was, this was a case study, very cool case study. This is an image of this individual's muscle fiber. And what you'll notice here is you imagine people that using anabolic steroids that are doing a lot of resistance training probably have much larger muscles than average people. Yes. And in some cases, even larger muscle fibers. And if you see this one, comparing it in your mind to what the slides looked previously, you could visually even see that there's more nuclei on these. And it's and it's much larger than average. So what we found is this is the fast twitch myosin heavy chain 2A fiber size. On average, interestingly, we didn't find that this elite powerlifter's muscle fibers were all that much bigger than an average person. You might think it's weird that I have a mouse and a rhinoceros here, but I'll show you why in a second. Much larger than a mouse or a rat, as you'd imagine, smaller than a larger mammal, as you might imagine. But we did find some outliers, which I don't know that have been reported in the literature. I've never seen them. And some of those outliers were actually huge. So we did find this fiber, for example, was much larger than the average rhinoceros muscle fiber and, about a third larger than the average human fiber. So, whether it's anabolic steroids, the lifting, genetics, we can't say, but this is just an example of how some of this physiology might show in, practice, might show out in practice. And a lot of the research is in animals, which I'll show you here in a second, but having a human participant like this was interesting. And then looking at the myonuclear domain size, you'd imagine that that big fiber needs a lot of nuclei. Well, yeah, a lot of the muscle fibers in this individual had much smaller myonuclear domain size, meaning more nuclei per volume than average. We didn't have it'd be perfect if we had some identical twins, which I'll also talking about in a second here, but we didn't have anybody like that to study as well. But again, that's an example in humans from our lab. But is it really that simple? If you just get more myonuclei, you can grow more, right? And it seems like linear and perfect and simple. Of course, it's not always like that. There's other things that we got to consider that people have been studying. One would be like the person or the animal's age, training status, other organelles responsible. So think about, what other organelles are important in muscle for growth. Probably things like ribosomes, which actually make the proteins, right? So that could be important. Epigenetics, which is a whole nother thing I won't even be able to talk to in this talk, but it goes beyond this. Things that can hang around after you've exercised. You can kind of reprogram these muscle cells, and then muscle fiber type. Genetically, people might be predisposed to have different types, or ability to shift might be different. And then some of this research was done by my colleague, Dr. Kevin Murak. He's at the University of Arkansas now. He did some of this at the University of Kentucky and then a lot of it at Arkansas. But what they found was in mouse models, they were looking at type 1 fibers and type 2 fibers. If you have satellite cells or these stem cells, these muscle stem cells, and you want to have the muscle fibers grow, type 1 and type 2, slow twitch, fast twitch will both grow with these stem cells around. But they are able to actually knock out the stem cells. So if you get rid of stem cells, you're not able to add new nuclei. What happens if you want to grow the type 1 or the type 2? Well, you give them, the stimulus to grow, let's say exercise or a different model in this case, but exercise them, or if we exercise and we had no stem cells, our type 1 fibers might not actually be able to grow that much or might not hypertrophy significantly at all, whereas the type 2 fibers might be able to keep growing because they might have that more flexible myonuclear domain. It could be related to, again, protein synthesis, or the needs of the slow twitch fibers are different. So that just shows it's not as simple as more equals hypertrophy, because in some cases you might not even need more to get hypertrophy. So if you're interested in this, people again been working on this for a few years, and it just keeps getting more and more exciting and cool. So there's more research to be done in this field for sure. So what are future directions? Um, I'm interested like epigenetics, genomics, things like that. This was a paper that we published. I was asked by Dr. Mirak to help collaborate on looking. It was just a pretty cool review article, but looking at this myonuclear domain idea, what we've learned before, where we're at now, and things to look at in the future. And one of the cool things is looking at myonuclear motility, or the ability of these organelles to move around inside the muscles and how that works. They use these proteins called kinesin and dynein, which are really similar in function, and they look similar to myosin heavy chains because myosin is a motor protein. They climb on these microtubules, which are little tracks that kind of just basically get created and then destroyed when you don't need them anymore. So the myonuclei can go places and do things. So that's one cool thing that I think is the next thing people are looking at. And then also, if we think about the myonuclei, they're not all equal. They don't all do the same thing. Some of them are special. Some of them have distinct purposes. So think about the myonuclei not like everyone's the same, but almost like a city where you might need police myonuclei and fire myonuclei and ambulance myonuclei and, teaching myonuclei, all these different ones. We've got ones responsible for repair and damage, maybe some for growth, some that are responsible just for maintaining where the motor neuron attaches at the neuromuscular junction. So there's a lot of things to study around that as well. So now next section is thinking about muscle atrophy. So growth, again, really cool, really interesting to study, but on the opposite end of that, atrophy is really important to study for a lot of reasons. If we think about atrophy, things that cause atrophy, aging, um, inactivity. Thinks about if you've ever been sick or injured, you might get muscle atrophy if you've ever had to have your arm in a cast. And one other thing that got me excited about this, like a long time ago, I've always been kind of interested in space and space travel. And you can imagine, in the movies in outer space, there's zero gravity, right? Microgravity. Um, there's not a lot of loading on your muscles and bones and tendons. So space flight and atrophy has actually been a real problem because now people are living and working in space. So this is a picture of the International Space Station. It's been up there since 1999, and people have been living and working, orbiting Earth ever since then. If you're up there for a couple of days, it's probably not a big deal for your muscles, right? You might have other cardiovascular or vestibular issues, but your muscles are probably fine. But if you're up there for weeks or months or years, muscle is going to definitely be affected. So long duration space flight can cause huge reductions in muscle mass and strength. Right now, there are ways to exercise up on the space station, and there's plans to exercise on the moon and things like that too. But they can't totally prevent this. Even with exercise countermeasures, muscle mass and strength is going to decrease a lot, and there's even a shift in your fiber type, so the quality of the muscles change as well. One thought that's a good idea is if you want to maintain muscle mass on Earth, what do you do? Resistance train, probably more high intensity exercise instead of doing long duration slow things. So that's what was proposed in space. And there's three exercise devices in space right now, the ARED, then they've got a cycle ergometer and a treadmill. And this can actually load quite a bit of weight. It doesn't have weight, obviously, but uses kind of this pneumatic system to load a bar, and you can do various resistance training exercises on it. Then there's kind of this cycle ergometer that you can again load up quite a bit of wattage on there, and then a treadmill that is not 100% your body weight, but tries to get as much as you can comfortably pulling you down onto it so you're able to run in space. And this is Suni Williams when she was the first person ever, I believe. Correct me if I'm wrong, but I think I'm right that was the first person to run a marathon in space, which is pretty intense. Running a marathon on Earth is hard enough, but imagine in microgravity and being strapped down to this treadmill. So there is the ability to do some of this stuff in space. It's really cool. But even despite these countermeasures, we're not seeing what we need to see. And over the last couple decades, there's been some studies that have looked at this where they've done studies looking at 6 months on the ISS, as well as a ground-based analog looking at bed rest, which you can imagine is similar to spaceflight, not quite the same, but as close as we can get on Earth right now, and looked at high intensity, more low volume exercise on muscle and bone loss, as well as cardiovascular function. This is just an example of somebody's training log. I don't think everybody was exactly like this, but just shows that they did resistance training at least 3 days a week, interval training at least 3 days a week, and then they like continuous aerobics, so they also did that a few days a week as well. Even with all this, though, there was still similar losses. I kind of just want to hit that home. Similar losses in muscle strength and muscle mass and endurance. So even exercise is not enough up there. We're on Earth. We're, again, designed here to live on Earth in 1G, one gravity. And so when we exercise here, we can maintain or grow our mass because it's good. That's what we do. But when we're up there, think about it. You might not be eating the same. There's other things going on. There's also radiation, coming from the sun that you're not protected by as well. So there's a lot going on. And there's even a shift in the quality, not just the quantity of the fibers, but the quality. And this was a review article that we wrote a while ago, just comparing a bunch of different studies. And the main goal to look at what this was what happens to fiber type in all these studies with unloading or microgravity or bed rest. And we always, most studies will show a decrease in slow twitch fibers and then an increase in a lot of these hybrid fibers. So it shows that there might be this shifting or this kind of, you know, changing of the proteins inside the fibers if they're not being activated or used like normally. So you'll notice a lot, your body's pretty efficient. It doesn't want to hold on to muscle mass if it doesn't need it. So it actually makes sense. Like if you are, floating and sitting, your body's like, why am I using all this energy to maintain this mass? I'm going to get rid of it. Well, we want to still keep that because you're going to land on the moon or Mars or Earth soon, and you don't want to, break everything because you're just so deconditioned. So we've got to almost figure out a way to, exercise the way, nutrition, maybe even some drug interventions to trick the body and keep our muscle mass on at least. Growing would be great in space, but if you can just maintain muscle mass and maintain your fiber type, that would be the goal. That's what people are looking at. So there's some future directions. Our lab's been looking the last few years actually at the tendons as well. So if you think about muscle, you've got skeletal muscle and then you've got bone. But what connects those together? Tendons. So we've done some initial studies on this, and we're about to publish a few papers on it. But looking at the effects of spaceflight on tendon collagen morphology and then the nanomechanics. So looking at basically how rigid it is. So if you can imagine, tendons are almost like rubber bands, right? If they get too stiff, then they can snap. And if they're too compliant, though, then they're not going to be very good at, transmitting that force. So they've got to be kind of right there in the middle in that sweet spot. But it turns out, just like anything with spaceflight, it does change the morphology and the shapes of and the ability for them to load forces. So again, we've just published a couple of things on the OSD, which is NASA's place where we publish all data public. So if you are interested in looking for data that's already published, to, analyze yourself or help collaborate with and do stuff, you can check out NASA's OSD. It's all public because this stuff was funded by taxpayer money and we want to give back to everybody. So those images are up there, and we're working on papers right now on that. And if you are interested in this idea of kind of open science or sharing data and stuff, there is a free class through NASA called TOPS. It's transformed to open science. It's got these, modules you can take on your own remotely and get a certificate for that. So really cool stuff going on with NASA, but we've got a lot coming up with that I'm excited about. And then the last section I want to talk about for this part of the talk is metabolism. So again, muscle metabolism I think of as like the foundation of everything. And one cool thing about skeletal muscle fiber type, or if we're talking about myosin heavy chain type, is it doesn't just predict speed, slow twitch, fast twitch, but also your maybe ability to perform for like more endurance or more sprinting type activities. So we'll talk about the metabolism part of that as well. This is a picture showing percent of fiber type distribution, and we've got the type 1, 2A, 2X, and all those hybrids. These are studies showing just average adults probably. What you might find if you're just walking around on the street and you wanted to measure fiber type, you'd find maybe 30% ones, 30% 2As, and maybe 30% hybrids. Very few probably 2Xs. So very kind of like I would say we say this is pretty untrained because you don't have a lot of one or the other, right? You can kind of do a little bit of everything, but you're not going to be really great at anything. And this is an example of a case study that we did a while ago, looking at somebody who was really trained. And at this time, they were pretty trained, in their career, an elite MMA athlete. They had a ton, almost 70% type 2A muscle fibers, which are those fast twitch, but they're not the fastest fast twitch. And the reason why maybe they had a lot of these 2As is the 2As are kind of good at, oxidative stuff, kind of good at glycolytic. They're more glycolytic, but they're kind of the best of both worlds. If you have really oxidative 2As, they can be fast and they can go for a long time. So thinking about MMA, or I imagine sports like soccer or something where you've got to run around or move around a lot and maybe have short bursts of speed, you might need these 2A fibers, But if we were to look at somebody else, how would this, potentially look with different groups of people? So one thing our lab's been looking at with other labs, some of our main collaborators, Dr. Andy Galpin, we've been working on this kind of stuff for a while, is how does fiber type maybe predict performance or vice versa, I guess. If you knew somebody's performance, could you predict their fiber type? So this just shows again some in this case this person was really trained. They had a lot of these 2A fibers and very few hybrid type fibers. So if we kind of take this idea and we look at a compass, maybe where we have less hybrids down on the bottom, south, more hybrids are north. And then we've got more slow twitch fibers west and more fast twitch fibers east. So imagine this is kind of like a compass. If we look at our recreationally active people, sedentary people, we kind of find them in the middle. They probably got more hybrids and, maybe a little more fast fibers, but somewhere kind of in the middle. We had that elite MMA fighter, a lot more elite fibers, so more fast twitch again, way less hybrids. Then we throw up a bunch of other activity people. We might find some recreational runners, some elite distance runners, way more slow twitch fibers and a lot less hybrids. Then we've got bodybuilders, elite sprinters, power lifters. They're going to be over in this more fast twitch fiber area. So this kind of shows where, again, if you take these people and you're able to get muscle biopsies from them and do myosin heavy chain, they're going to fall probably in a certain quadrant of this. But how much of this is genetic? Like if you just pick somebody off the street and they could be a good sprinter, but they're not doing it, would they potentially have like a super fast twitch or super slow twitch fiber type? And that's, the question of nature versus nurture. Well, let's just like, I'm gonna, I guess, spoiler alert, but let's say nature and nurture. So we all know that it is training and diet and exercise and all that stuff super important, sleep, everything, caffeine, whatever, all that nurture stuff you can do, but the nature stuff you're kind of born with. But how much is, how much of that nature is related to fiber type? So we wanted to kind of study that and look at it. And a lot of other groups, again, have done this. There's been twin studies going on for decades that are getting really cool data on this as well. But we know that your ability to adapt to anything is going to be partially genetic. Some people, I hate those people, but they'll just go in a gym a couple days later and come out and you can already tell they're jacked, right? Most of us aren't like that, but let's figure out how much of our ability to adapt is genetic. So if we look at this on an equation, we can say your total variability of a trait would be VT in this equation. Your genetic variability plus your environmental variability, which would be like exercise, nutrition, all that, plus some measurement error, we're always going to have that. It'll give you that total variability of a trait. Um, so our idea is how much of your muscle fiber can adapt to exercise training. Like if you're given enough time, could you totally shift your fiber type, fast twitch or slow twitch, or whichever way you're going for. We got kind of given this really interesting example. I'm not going to talk about the background now because I'm going to talk about it in the next section of this talk, but we got um, access to these twins who had 30 years, so 3 decades of different physical activity history, where one trained to become an elite Ironman and the other was just kind of an average Joe. Not super unfit, but not an elite Ironman. So they had totally different physical activity histories. We wanted to look, so what are the limits? What if you just kind of lived a normal life or you kind of went for it for 30 years? What could happen potentially? This was a graduate student that I worked with colleagues at Cal State Fullerton. So Katie Bathgate led this, and then Dr. Andy Galpin was the PI on it, and I got to help out a lot. But we looked at the muscle health and performance in these monozygotic twins. So those were identical twins with 99.99% the same DNA with 30 years of different exercise habits. And what do we find? Again, we were looking at that Ironman triathlete versus the average Joe. Stuff you probably imagine. Less body fat in the Ironman triathletes, less cholesterol, lower blood sugar, lower blood pressure, and even a 30% higher VO2 max, which is pretty incredible. Like you can definitely train and increase your VO2 max, but 30% is pretty good. I mean, if we break that down, it's 1% a year. I'm sure it's not linear like that, right? But just shows that you have the ability to adapt. Um, but what about muscle fiber type, right? Do they just have the same fiber type? You'd imagine these identical twins, all things considered, should have been born with the same fiber type and same ability to adapt. So what do we find? This shows it's similar to that graph from a few slides ago, but showing the percent muscle fiber type distribution. Type 1, 2A, 2X. This is the untrained twin here in gray. So you got total hybrids, up about 30%, fast twitch up about 40, slow twitch a little under 40, right? So pretty average. This is what we saw in those other studies. This is what you can imagine. So now think about where would you think that trained twin is, that elite Ironman athlete? More type 1, more type 2A. Well, we figured more type 1 also, but we figured maybe a few percent more, right? Probably from training. Well, we ran it and we got some incredible results. So we ran more tests, and we kept coming up with the same answer. We found almost 55% more slow twitch, those oxidative fibers in that elite Ironman athlete than the identical twin. And a lot more or a lot less hybrid fibers, right? So a lot more type 1 slow twitch, a lot less hybrids. This is more than we thought we would find. And I think that this shows that maybe not everybody, like maybe I can't shift my fiber type 55%, but it shows the ability in humans is there. And if they can do it, then some people certainly can. So our abilities to adapt are probably in some people more than we even think or know because we haven't done these studies. Now, let's get that study going where we get some people, training for 30 years and then some identical twins not to. We're going to have to go and find some of those. So if you are interested in this research, there's a lot more to be done with the twin stuff. So if we throw those twins up on that compass that our lab made up, we've got the untrained twin up here with the more hybrids, a little more fast twitch fiber type, right? And then that elite endurance athlete way down in the slow twitch fiber area, way less hybrids. So we would say like kind of way more trained for that specific um, performance variable, which would be going longer distances. All right, so that's kind of again, there's a lot more to do. We don't have. I've had students ask, well, what about rugby players and what about water polo players and what about this? Like, we don't have a lot of data on a lot of different athlete types, so for various reasons. But if you're interested in that, that's definitely an open area of research. So some of the stuff that our group and our colleagues have been getting in future directions is looking at like the link between the gut microbiome and metabolism, specifically in people who do ultra endurance events like ultra marathons. We've done some work looking at the gut microbiome changes during a marathon and then how different marathoners might have different gut microbiomes than, the average adult or that average Joe again. And it's probably adaptations and things that probably are going to help you during these exercise bouts. So this gets even deeper. It's not just microbiomes, not just one thing. These are all the different types of bacteria and viruses and everything living in your gut, and it's kind of like its own ecosystem. So imagine it's almost like a rainforest in there, and you've got to have certain amount of diversity is good. But if you get the wrong bacteria, it's bad, right? And if you get some of the right bacteria, it could even be better. So people are trying to figure out how the gut microbiome influences exercise performance, but also how exercise performance or exercising or physical activity can influence your microbiome. This was some of our groups. We had a great group this last few years that have gone back to the Western States Ultra Endurance Run out in uh, Tahoe area that goes down to Sacramento. It's a 100-mile race and have been able to see some really cool stuff. So this was our group in 2023 that was there. Just published the first set of a few different articles coming out, but a lot of looking at cardiovascular health and metabolomics, kind of the next thing people are looking at for that. So you can imagine it takes a lot cardiovascularly to do this, and a lot of muscle damage is accrued during 100 miles, especially when it's like a net downhill because of all the eccentric loading. So studying that is really interesting. The good thing is it seems like with proper training, you can safely do something like a 100-mile race. And probably everybody has different adaptations or has a different ability to adapt to that. And that's what we're looking at. Want to try to make athletes be able to do this as fast and as safely as possible. All right, so I got one more section for you before this is just too long of a talk, right? But I want to just give you a background story. This was a story from a few years ago, but I want to talk about how does a research idea become reality? Because I'm sure a lot of you have research ideas and they just seem like ideas, right? Just on your paper. But how can you make that a reality? And I'll show you it might take more time than you think, but it might happen. So this is that study, right, that I showed you, the twin study that Katie Bathgate led on monozygous twins. We found that crazy 55% slow twitch fiber. This one figure, right, encompasses everything, but it seems like, wow, great, one figure, check. Well, it's a little more than that. I'm going to talk to you about how does that one figure kind of become a reality from an idea. And that was a figure from a paper, so a peer-reviewed article. And if any of you are in the process of writing a peer-reviewed article or in the publication process, you know it is a lot of work and it's really rigorous and it's time consuming and sometimes you might not think it's worth it, but I'm going to hopefully make you think it's worth it by the end of this. So part one of trying to make your research idea become a reality is serendipity. A lot of this stuff just happens. So serendipity, we can look at the um, definition here is the occurrence or development of events by chance in a happy or beneficial way. So some people say a happy accident. A lot of science just happens, on the back of a napkin at the restaurant or the bar, right? Or talking to somebody while you're going for a jog or bumping into somebody in the hallway. Serendipity. Um, this happened when I was at finishing up my PhD. I had like a year to go, and I was, in this summer, I didn't have funding to continue work. And I was like, what am I going to do with my summer? And I ended up having a colleague and friend, Dr. Andy Galpin, who was just luckily starting up a lab out at Cal State Fullerton. And I ended up spending a few months with him, helping him set up a biochemistry lab to do fiber type and stuff like that. He had really great access at Fullerton to athletes through the Center of Sport Performance. And I was just really stoked to help him out. And summer in SoCal is not bad. You could surf as well. So I was talking to a graduate student there one day and we were just isolating muscle fibers, which is super time consuming, but it's fun because you can talk. And she was saying like, oh, my dad's an Ironman triathlete, and I was like, that's cool. And we're just chatting about endurance exercise, and it's like, hey, he's a twin, and got into that. I was like, that's interesting. And I had just read some studies from there's a big study still going on in Finland on identical twins. When she said that, I was like, oh, I bet is your uncle an elite athlete too? And she said, no, he hasn't done any exercise since high school. And I was like, what? Whoa, it's a long time. So you're saying you basically have somebody who is an elite athlete who's been training their whole life, and then somebody who hasn't, and they have the exact same DNA. And she said, yep, and so that got us thinking, and we started being like, well, maybe this could be a case study that could potentially be some students' graduate project and probably get some papers out of and learn a little bit about physiology. A lot of times you need a huge sample size, but when you have something this interesting, a case study or a really small sample size can give you some good information. So we started reviewing the literature and looking at things we might want to do if we were able to study these twins. So part two, this is where collaboration comes in. So remember, I'm still a student, minimal money funding, well, no funding really. But I did have people to collaborate with. I had friends and I had people doing cool stuff. So this is where you've got to work jointly on an activity to create something. And a lot of times you get something good out of it. Everybody gets something out of it. And that's usually the goal of a good collaboration. So in this point, we're in spring 2015. So I wanted to go back. That started in 2014. All right. So now we're the next year, 2015. This is where we built our team. And Dr. Andy Galpin was the lead PI on this. We worked with Dr. Eddie Joe, a bunch of collaborators on this, and everybody had their own different experience and expertise. That summer, this is a picture I'll let you know. This was basically where my parents were at the time. Dr. Galpin and I had spent a few days out there writing IRBs, trying to get funding for this, but we didn't have a bad view. So there's an ocean view. Our work ended up paying off. We got some funding, and we were able to start planning the logistics of getting this group of participants in there to look at that fall. So, we're over a year now of working on this. And by this time, I had graduated, gotten another job. My current job really is I started as assistant professor and I was continuing to do this research with them. So part three was perseverance. So this is, you got to do something despite it being difficult or delayed or difficult or delayed. Or we're already a year and a half in, right? We're in spring 2016. Finally, we got to do some data collection. And this data collection happened all over Southern California. We did DEXAs and performance tests and biopsies and all kinds of really awesome things on these twins. And that summer, got initial data analysis out, finally shared the first bit publicly that July. So we're a couple years down the road. And then I'm newly minted assistant professor. You can see here my hair is a little shorter. This was 2 in the morning driving into Southern California from San Francisco. Is not a short drive, but I had our samples and was greeted by Dr. Galpin's dog at the door, and we got those in the freezer and then continued data analysis that next year. So now we're in spring of 2017, all the way to that fall, ran more analysis, finalized data sets. This is us running gels for muscle fiber type. We got some of our students to come in, did some practice biopsies on them. It was a great time, but we're years down the road, right? Remember when I was like, make a research idea a reality? The point here is it takes a long time. And finally, in that spring of 2018, we got our paper ready to submit, and we submitted it in March, and we got a quick rejection back. So imagine working on something for 4 years, pretty stoked on it, right? You think it's good, boom, rejected. So what do? We made edits. We to that article, we went to another journal for peer review, and in May we got major revisions, and then in July we resubmitted with edits. So this is that idea of perseverance, that review, revise, repeat, keep going around until it gets perfect for the reviewers and for everybody else. Finally, in July, we got accepted for publication in 2018. So the point is, that's a 4 year process. And then, boom, we're done, right? Check. No, you're not done. Once you publish it, it's great. But if you don't talk about it or, talk about it at conferences or publish it in other places, then nobody's going to really know about it. So, part four of this whole thing, and a really important part of science, especially nowadays, is communication. If nobody knows about your research, it might as well not exist. And then you've wasted all your time and energy, right? And then if nobody knows about it, then they're not going to fund it in the future because they think it doesn't exist. So it's up to us as scientists. We weren't trained to be communicators mostly, but we got to try to communicate, whether that's social media or our friends and family. We got to try to get that out there. So backtracking to summer of 2016, remember I told you Dr. Galpin and Katie presented this at the National Strength and Conditioning Conference. That was the first bit of data out on that. We presented even more data in fall of 2016 at the American College of Sports Medicine. And then after it had been out for a little bit, we got kind of blown up. We ended up talking to some people at Men's Health magazine, and they wrote a whole article on Dr. Galpin's lab, and then a bunch of other opportunities presented themselves. So we presented this at the National American College of Sports Medicine. That finally that article came out that July, and then a bunch of other smaller kind of media people contacted us, and we were able to get that information out. Turns out that identical twins is kind of like quote unquote sexy science. So if your science has anything in it, even if it's like aging or longevity related, sport performance is pretty sexy. But this, people like to study twins because it's kind of interesting because we do know they have the same DNA. So that's a great model, right? But it's also pretty rare. And then that fall, I thought we were done at this point. We did all our communication, right? So I'm sitting at a conference down in San Diego, and the keynote speaker is Dr. Mike Joyner, who's out at the Mayo Clinic. And this is a picture I took from the back, and I'm sitting there in the back. Probably it was like 8 a.m., having my coffee as you do at conferences. And, I was excited and stuff. But then heard my name and I was like, whoa. I looked up, and that figure you can see there is the figure that I just showed you, showing the 55% more slow twitch fiber. And Dr. Joyner was talking about our twin study on here. And then I got real excited. I was like, oh, that's pretty cool. So afterwards, I went and introduced myself and then had coffee, and we talked about writing a grant. So we started working on writing a grant together. So this is where opportunities come up. Some opportunities might not work out, some might lead you to other opportunities. Some might work out, brilliantly better than you could even think. So opportunities is a set of circumstances that makes it possible to do something. So by doing the research, communicating it, you might get some opportunities that you never would have gotten before. So we had that peer-reviewed article. We submitted some grants with the Mayo Clinic and a big collaborative team. It was really great to work on these grants together. Um, Submitted that. We got rejected, unfortunately. We're trying again. We're always thinking about follow-up studies for this. So if anybody's interested in twin studies, there's stuff going on. But yeah, some really cool stuff came out of this one conversation that I had with a graduate student in 2014 when I was a graduate student, and it led to this great, almost like 10-year long thing that led to even more things that I couldn't imagine. So here's that timeline recap just up till 2018 because it's like continuing on, I would say. It's not over, but the research idea started a long time ago, and there's just a lot of these things. Right now, looking back, it looks like, oh, it was all planned. None of this was planned. So another thing is your research might be serendipity. It might turn into the 10-year thing and it might end up becoming a one paper and that's it. You never know, right? So thinking about this and just going back full circle is as you're coming up with idea, try to think about things connecting the dots, integrative view of things. So we look at an integrative view of muscle physiology. You might look at an integrative view of the environment. You might look at an integrative view of a different system of the body, but the whole body, all the way down to the molecular level, is all connected at these different levels. And it does ultimately help you predict or change human health and performance. So some take-home messages is that, muscle cells are really, really adaptable. They're pliable, they're plastic, right? We know that myonuclei can drive muscle growth, but probably not the only thing driving muscle growth. It might just be one piece to this bigger puzzle. We know space flight decreases muscle size and quality, so space is bad, but we have to basically have humans living and working in space if we want to keep exploring. So we've got to figure out a way to make that work out. Fiber type can influence exercise performance. So we can see that just looking at this compass, if people that are, have a lot of slow twitch and a fewer hybrids are probably going to be really suited for those kind of endurance-based sports or those really long distance type running sports. Whereas somebody that has a lot of hybrid fibers and maybe not too many slow or fast fibers, they're kind of right in the middle where those average Joes are. So I don't think that's bad, but I think that means that they're poised or ready to go, right? You can train and probably shift one way or another. And then this whole second section was all about research, right? It kind of just want to talk about that research is hard, but I think it's worth it because you're going to get a lot out of it, not just that paper, that line on your CV. You're also going to make friends, you're going to make connections, you're going to do stuff that you never thought you would have done because you went down these rabbit holes. But research is hard because you're on the fringes of what we know. If you're in school and you're asking questions and nobody knows the answers, you're in the right spot. Like that's what we're all doing. Like if you don't feel a little bit dumb doing this stuff, then you're not doing real science, I think. So even the people that win Nobel Prizes will tell you, yeah, they feel dumb all the time, probably. Um, oh, I do. I'll say I do sometimes, I do a lot. So you got to embrace situations, even if they might be uncomfortable, that lead to the serendipitous environments. A lot of research and science is going to require collaboration and for sure perseverance. Anybody that's been in this longer than 5 years will tell you it doesn't get easier. You might get more opportunities and stuff and bigger things will happen, but you're always going to have perseverance. And then communication is going to really open up a lot of opportunities. I mean, go to conferences, go to even conferences on remotely. You don't have to go in person sometimes. Be active, ask questions, talk to people, and try to just learn. Be a sponge, especially if you don't know what you want to do, but you like research. Just put feelers out everywhere. Try to join a bunch of labs, and something will definitely happen. You'll find what you like. All right, so that's just my little pump up talk and our research talk. I want to thank everybody that's helped with Muscle Phys Lab research. These last 10 years have been great, and I hope we'll have another 10 and 20, 30, 40 years and keep the lab going. And that's going to depend on everybody and all of us working together. So if you are interested in joining our lab or hanging out and coming to visit, you can always contact me. My info is here on the left. Our website's on here. We have social medias and all that. But I'm happy to entertain any questions on social media or email. And I hope you enjoyed this talk. Thanks for listening.